So, picture this. You've just spent hours, literally hours, memorizing beta-glucose 1,4-glycosidic bonds. You walk into your exam, you write the seemingly flawless paragraph on microfibril tensile strength, and then results day comes around, and you score a 0 for that question. No way, right? Why does that happen? Well, it's because you fell right into the examiner's trap. Most students lose really easy marks on the cellulose structure and function topic, but after this explainer, you absolutely won't be one of them.
You really have to ask yourself honestly, are you falling into the classic terminology trap that constantly shifts these grade boundaries? The brutal reality of Cambridge A-level biology is that having perfectly memorized facts, it's completely useless if you apply the wrong terminology to the wrong context. You might know literally everything there is to know about plant cell walls, but if you don't know exactly what the examiner is actually asking for, you're leaving marks on the table before you even pick up your pen.
And hey, this isn't just my opinion. The Cambridge Principal Examiner reports stated incredibly clearly. They say candidates consistently confuse key terminology, always define terms before using them in extended responses. This is the ultimate proof of why normal rote memorization revision just completely fails. The examiners are explicitly telling us right here that students are bleeding marks simply because they don't lock down their definitions before diving into their analysis of cellulose structure.
All right, let's dive right in. Section 1. Core concepts and evaluating evidence. So, the academic benchmarks for Biology 9700 require really logical, structured reasoning. The assessment framework here prioritizes analytical depth over superficial breadth. In other words, examiners do not want a brain dump of everything you happen to know about cellulose. They want to see if you can handle and evaluate information systematically. A really crucial concept here is contextual specificity, and this is the #1 warning from those principal examiner reports. Contextual specificity is the absolute requirement to explicitly link your biological terms and your units to the exact scenario provided in the stimulus. Generic answers about plant cell walls, that's a fastest way to score a 0. If the data is about the structural integrity of a celery stalk, your answer must be about that celery stalk, not just a generic textbook plant cell. So how do we actually guarantee we hit that level of specificity?
Well, that brings us to Section 2, the method mark strategy.
In Cambridge exams, marks are frequently awarded for the process, not just your final conclusion. This strategy is all about showing the examiner your analytical framework, step by step. Here is the standardized procedural sequence you need to safely navigate and conquer literally any cellulose question. First, deconstruct the question. Ask yourself, what cognitive domain are they testing? Second, extract relevant data straight from the stimulus tables or graphs. Third, select your analytical framework. Fourth, execute your analysis with incredibly clear notation and working. And fifth, present your final results using the correct terminology. If you follow this exact sequence, you're building this amazing safety net of method marks, even if your final calculation is just a tiny bit off.
Now pause for just a second. In a moment, I'm going to show you the hidden examiner clue that changes your answer completely. It's a clue that basically flips the entire mark scheme. But to make that clue obvious, we first need to master the top scoring framework that examiners actively hunt for when grading your papers.
And here is that ultimate top scoring framework. Every single time you write an extended response on cellulose structure and function, you've got to structure it exactly like this. Step 1, state the biological concept. Step 2, provide evidence or data extracted directly from the stimulus. Step 3, apply scientific reasoning to connect that concept to the data. And step 4, finish by mirroring the exact examiner wording used in the question prompt. This loop forces you to be specific. It proves to the examiner, without a doubt, that you are operating in that top mark band.
Let's put this into practice with Section 3, Interactive Challenge, Spot the Error.
I want you to mentally pause for a second and actively predict exactly where this student lost marks based on what we just learned. Take a look at the flawed answer first. It accurately describes cellulose, sure, but it uses really generic terminology, it misses the specific data units from the table, and it completely lacks a contextual connection to the plant cell wall stimulus that was provided in the paper. It's essentially a textbook answer to a practical question. Now compare that to the top band answer. It perfectly executes our framework, concept, evidence, scientific reasoning, and examiner wording, all explicitly tied to the unique context of the experiment. That, right there, is literally the difference between a C and an A star.
Moving on to Section 4, the ultimate checkpoint, data analysis. Bridging the gap between biological theory and experimental data means you absolutely need to know your statistical tests. A massive part of handling and evaluating information in the syllabus comes down to interpreting data from practical investigations on plant cell walls. So here is your ultimate cheat sheet.
Use a t-test when you're comparing the means of two different sets of data, like say the tensile strength of two different plant fibers. Use the chi-square test when you're dealing with categorical data, and use correlation tests when you're looking for relationships between two continuous variables, like cellulose concentration and cell wall rigidity. Seriously, memorize these use cases.
But statistical analysis is really just the beginning. Top-scoring candidates consistently demonstrate critical evaluative thinking. This means you must explicitly identify limitations in the practical apparatus. You've got to question the assumptions made during the experimental setup. You need to consider alternative interpretations of the data. And most importantly, you must justify every single decision or conclusion you make with hard evidence from the text.
And that brings us back to our open loop. What was that hidden examiner clue I mentioned earlier that changes your answer completely? Well, it's the context. Context is king. The clue is always buried right there in the introductory paragraph of the question, which is the part most students just completely skip. Perfectly memorized biology facts score absolute 0 if they aren't explicitly tied to the question's unique context. If the stimulus is about an experiment identifying anomalies in cellulose digestion, your answer cannot just be about the structure of microfibrils in general. It has to be about the anomalies in that specific digestion context.
All right, let's look at some final exam-focused takeaways.
Let's wrap up this explainer with a quick recap of the most high-yield strategies we've covered today. If you apply these steps, you will completely transform how examiners view your paper. Here are your 4 pillars for mastering cellulose structure and function. First, always define your terminology before using it in an extended response. Second, apply the method mark strategy to show your analytical steps clearly. Third, obsessively check your units and ensure your answer has contextual relevance to the stimulus. And fourth, know exactly when to execute the correct statistical test.
But before we go, I need to leave you with a final, kind of provocative thought. You can master all the cellulose concepts, you can absolutely nail the contextual specificity, and you can calculate the perfect t-test. But what if your perfect, top-end answer is completely ruined just because you misread one single hidden command word at the very start of the prompt? Think about it. If the examiner asks you to evaluate, but your brain reads describe, you are going to lose every single mark for that question, no matter how brilliant your biology is.
These command words literally dictate the mark band you're placed in. If you want to know exactly how to decode them so you never fall into that trap, you have to watch our next explainer. Subscribe to our YouTube channel, Ace the Exams, at Ace the Exams IGCSE to excel in your Cambridge examinations. Check out the next explainer right now to master those command words, and we'll see you in the next masterclass.